Please welcome Jerome Etwaroo, University of Victoria.
Hi, everyone. On behalf of the University of Victoria, I would like to introduce Dr. Karolina Valente, founder and CEO of Voxcell BioInnovation. Karolina is a brilliant innovator, and I am here because of Voxcell is addressing drug discovery and enabling personalized medicine in 3 different ways. I think why Voxcell for the following was Venture of the Year. There are 3 reasons. 1, they have an amazing team. They have a team that is cohesive and very skillful. Number 2, they are meeting every critical milestones in their journey. And thirdly, they are solving a real problem. Their technology is solving a real problem. And I'll give you a quick story. Earlier this year, I lost a family member, a matriarch in our family. She was diagnosed with cancer, and unfortunately, they couldn't find a perfect drug candidate to reverse the disease. Unfortunately, we lost her, but Voxcell BioInnovation is going to be changing that, because they are the ones are going to change the way medicine works, personalized medicine. And that's why I'm excited to be here, because Voxcell is the future of health. Thank you.
Breaking the wall of drug testing in oncology. Karolina Valente, Voxcell BioInnovation.
The drug development process is the process in which pharmaceutical companies take drugs from an idea to hopefully the market. But let me tell you something shocking in the case of oncology. This process has a failure rate of 95%. It takes about 15 years and more than $2 billion. And in case of testing, this process starts being tested in the lab in the preclinical development stage, and the most promising drug candidates are then selected to be tested in humans. The problem is most of them fail. And they fail because we do not have platforms in the preclinical stage to behave as a human sample and to predict human behavior until now. Voxcell is developing human-like cancer tissue models using bioprinting technologies. These tissue models are going to be used as a platform to screen the different drug candidates and to select which ones should be moving forward, increasing the odds of drug efficacy. Let me show you how we do that. As I said, we are using bioprinting, and that has been 100% developed by our team. We combine a high-resolution bioprinter together with a vascularization software and proprietary bioinks, that is our printer's cartridge. And the result is a real 3D tissue that contains real cancer cells and a human-like vasculature. And the idea is that pharmaceutical companies will inject the drug candidates in the vasculature and will select which drugs should be moving forward by analyzing how these drugs are interacting with the cancer cells. The goal is to eliminate the inviable drug candidates earlier, allowing time and money to be put in the drugs that are more promising to work. Because we developed our own technology, we can create very customizable tissue models, different cell lines. On top of that, our tissues are viable for 3 weeks, unlike anything else on the market, really cutting-edge technology. And they're highly translatable, which means that our goal is to mimic human behavior. And because they're going to be used as a screening platform, we actually do not require FDA approval, de-risking the technology and accelerating the time to the market. Our customers will be mainly pharmaceutical companies, and these products will be sold as consumables starting next year. So it's really a B2B platform with standard tissue models and also customizable tissue models. And it's driven by partnerships with pharmaceutical companies. And there has never been a better moment to be putting this technology on the market, because the FDA and the EMA are really driving towards innovative technologies that can can eliminate animal models. That is that step that comes right before clinical trials. So between Voxcell's technologies and clinical trials, you have animal models. So if we can decrease that number of animal models, and this is exactly what we are doing with our technology, is really behaving as a human sample. We spend a lot of time talking to key opinion leaders out there. The company is about 3 years old. We seek a lot of feedback. And as a consequence, we actually have 10 pilot projects, and we are expecting to generate $1 million in revenue next year and 10 in the next 3 years. Voxcell has raised 2 oversubscribed equity rounds and also $2 million in grants and awards. And right now, we are validating our tissue models, really getting ready to put on the market and getting ready for our Series A next year. And none of this would have been possible without an amazing group of people that are growing this company together with me. We are 13 full-time employees. We have a 6,000 square feet facility in Canada, and we are creating the hardware, the software, and 3D printing these tissue models to be sold as consumables. Our management group has expertise from 3D printing, tissue engineering, and business development. And right now, we are also having a scientific advisory group that is providing a lot of support for us with Dr. Mark Pigram, that is professor of medical oncology at Stanford, and Dr. Deacon, that used to be the former head of microphysiological systems at GSK. Our mission is to accelerate the drug development process because drugs should be getting to the market sooner, into the hands of people that need them. And Voxcell will make sure that this happens. Thank you.
Wonderful.
I have a sample here.
Oh, wow.
Not a real sample, but it mimics. And the real sample is about 5,000 times smaller than that.
Okay, that's impressive. Let's start with the question. Okay, we have 2 questions here in the first row. Oh, okay, another one. Okay, let's start with the gentleman first. Okay. Just please.
You said that your organoids or printed organoids are viable for 3 weeks.
Yeah.
And what differentiates you really to the other technology is the vascularization.
Correct.
That means you also have a blood supply for 3 weeks?
That means that the drugs can be injected through that blood vessel-like structure that we are populating with endothelial cells.
Yes.
So yes, yeah, and we are actually the next level than organoids. We are much better than they are.
But do you have drug supply? Do you have blood supply for 3 weeks?
We have perfusion of whatever you want to do. So when we are talking about the drugs, you are doing blood supply plus drugs, or you're doing a combination. So yes, for 3 weeks.
You keep this perfusion on for 3 weeks?
For 3 weeks, completely viable with more than 80% cell viability.
Chas, please.
This is beautiful.
Thank you.
One of the challenges we've got treating solid tumors is that existing drugs don't get to the center of the tumor.
Absolutely.
So do you have any handle on that with this assay that you've been
Yeah, so the tumor microenvironment, that's the most difficult part of the cancer environment, and that's what we are mimicking with the vasculature, actually, because the vasculature in the cancer environment is very different than the healthy one. It has dead ends, it's very disorganized, and that's the key part of the technology that we are developing with Voxcell. And on top of it, the tissue actually is mimicking the chemical and mechanical properties of that human sample. So we are mimicking all the basically parameters of the tumor microenvironment. Thank you.
Okay, we have a next question here. I think it's Angelika.
Yeah, thank you very much. A very highly demanded technology, I think. Could you please spend a few words on your proof of concept, just a few words on
Absolutely.
cells, indications?
Yes, 2 stages of validation that are currently being done right now with our first tissue, that is the breast cancer. So stage 1, validating with drugs that are already on the market and which we have human sample results that we can compare. Stage number 2, validating with drugs that failed in humans due to efficacy, to illustrate that our platform can trigger failure earlier on. When we talk to pharmaceutical companies, that's what they want. They want to fail sooner, and they want to fail faster. And that's our main focus.
Thank you. The next question will come from here in the first row in this block, please. Thank you.
Yeah.
If you want to model the tumor microenvironment, surely the immune cells are also needed. I mean, are there any plans to actually elaborating on what Frank said, to incorporate that?
Absolutely, that's the next stage of our tissue model. So we're now working with BC Cancer Agency, which is one of Canada's large cancer research center, really focused on the development of immunotherapy together with our samples. So that would be the next level of enhancement in the pipeline for 2025. Thank you, Martin.
Thank you for the qualified questions so far. We still have 44 seconds, so Stefan, please, here in the first row. Yes, thank you.
In the documentation, you said that your tissue is at TLR4, yeah, compared to the other ones. When do you reach TLR 6, 7?
We are right now TRL around 5, very close to 6, and our goal is to have to 7 in Q1 to Q2 next year. So we are
What's
quite
the major
close.
challenge to go there?
Major challenge right now has been, I would say, really the perfusion is the most challenged part, is keeping that drug flowing for 3 weeks with maximum cell viability, which we just passed this major challenge around October. So right now it's just validating the drugs.
Wonderful.
Yes.
Soft landing perfect on 0. Thank you so much.
Thank you.
Very happy to